Okay, let's continue with microtubules. We have finished the intermediate filaments briefly. This is the second cytoskeletal element that we are going to talk. And let's check how it is localized in the cell. So unlike the other intermediate filaments, which is expressed, which exist anywhere in the cell, they are localized in the center of the cell. So all the microtubules in the cell, they originate from one single center. This is the first thing. They originate from centrosome. They are the thickest elements. So see, it is the thickest that we have seen. And 3, they are dynamic. So the intermediate filaments that we have seen in the previous video, they are very strong, they are very resistant, but they are not dynamic. They are mostly stationary. Once you have the intermediate filaments, you have them all along. On the other hand, the microtubules, they are dynamic, they polymerize and depolymerize continuously. And this is the normal life of the microtubules. So check the microtubule structure in the cell. It is all around, it is everywhere, but they all center from the centrosome here in the cell, and wherever they go, they start from here. Okay. The centrosome is the name that you have been seeing since the high school, but we don't call it centrosome anymore. Actually, we usually prefer to use this name, microtubule organizing center, MTOC. The microtubule organizing center in the non-dividing cell is only one, and it's usually called the centrosome. During the cell division, it is replicated, not even replication, it becomes 2. So all the microtubules depolymerize, they form the mitotic spindle here, and these mitotic spindle, they move in opposite directions. Okay. And the only place that the microtubule organizing center exists is not the centrosome. Actually, they exist just beneath the cilium too. So unlike you have learned in high school that a cell has only one centrosome, we're going to say that a cell can have several microtubule organizing centers, 2 if it is dividing, and much more if it is having cilia or flagella. Of course, we are still not sure about the importance of the microtubule organizing center or the centrosome, because plant cells, they are doing perfectly okay without them, so we are not sure what's really happening. Now, first see the structure of the microtubules. Of course, it has units. These are the monomers, alpha and beta. So the monomer consists of 2 globular proteins. Remember, the intermediate filaments, they consist of a filamentous protein, which is dimerizing. In this case, it is not filamentous, it is globular, and it consists of 2 different proteins, which gives a polarity, a directionality of the microtubule. So the only reason that the microtubule is polar is that there are 2 different proteins. Now here, where the beta faces is called the plus end. Now where the alpha faces is called the minus end. Again, this minus and plus has nothing to do with electrical charge. It is totally unrelated. It's about the growth. The plus end grows faster. That's it. It's the biological term. Now, several alpha and beta come together, line up, and they form a protofilament. The protofilament itself is not too much stable. Okay? It has to be stabilized with the help of other protofilaments. So altogether, there are 13 protofilaments come together. They are attached to each other with non-covalent bonds, and they form this microtubule. Now, the attachment of this to this and this to this in a protofilament, different dimers is totally non-covalent, and again the side-by-side interaction of the unit, the subunits, is also non-covalent. But again, because there is a very large number of non-covalent interaction, at the end it becomes a very strong structure. Microtubule is one of the strongest things. They just break under stress. That's why I don't call them strongest one. And inside the microtubule, in this lumen, it's hollow. There is no monomer or something. It's empty. Okay, and this is a very nice electron microscopy picture of it. Very successful. Okay, well, this minus end, it grows slowly, and this plus end, it grows fast if you do an experiment in vitro. In vivo, it doesn't even happen that way because the minus end is not free. The minus end is here attached on the microtubule organizing center, which is also called with the old name centrosome. Now, centrosome consists of a pair of centrioles. They are localized in the vertical position, and they are actually pieces of microtubules. And around the centrioles, there is a gel, so it's covered with a gel. And we really have no idea what the pair of centrioles or the gel is doing, because what really matters is that there are gamma tubulin ring complexes here. Okay, that's the functional stuff. So from each gamma tubulin ring complex, a microtubule is growing, and from this microtubule, the minus end is attached here, and the plus end is free to grow. So what happens if you remove the pair of centrioles? I don't know. But when it is replicated, this pair of centrioles is also replicated. So it's a star-like shape. And if you check the confocal microscope, it looks like this. These are the pair of centrioles, and these are the gamma tubulin complexes and the microtubules growing. Now, these microtubules, they are seated here. They are going to have their root on the middle of the center here. Wow. I can move it. But this plus end is free to grow. Now, I can say that the minus end is center of the cell, and the plus end grows to the periphery, which is the plasma membrane. But it doesn't mean that this microtubule is going to grow until the plasma membrane. Maybe it's going to grow. Maybe it's going to start to shrink. It can grow, this can shrink, this can grow, this can shrink. This is what we call dynamic. I mean, the size can increase and decrease, increase and decrease again. Anytime with the using energy. This is also what keeps the cell reactive. This is why the cell can react to the, to the stimulus coming from the outer environment. Okay. To the periphery, they can grow and come back. This is very useful, especially if you're going to push the cell content to a direction. By the way, centrosome is always in the cell center. It somehow, with the help of the microtubules, finds the center, and then it grows. Imagine that the cell is going to grow to a direction. Maybe there is a food source there. Maybe it is going to eat a pathogen. Maybe it's a growth cone of the neuron. Maybe it has to grow this direction. There are some microtubule capping proteins which are localized just beneath the plasma membrane. Now, these microtubules, because as they grow, they attach here and they stabilize, they don't shrink, they continue growing. Because they continue growing, they become stable microtubules. And this way, the cell changes shape from round to an elongated cell. Now, imagine a developing neuron. It has to push the developing axon to some directions, and this pushing is possible with the help of the microtubules. A very long journey, it can pass from one half of the brain to the other half, it can go from one side of the brain to the other side, and this long journey can only happen with strong elements like microtubules. You cannot do that with actin. Actin is too thin for them. And there is a very nice event, dynamic instability. Okay. is the growing and shrinking of the microtubules, how they grow and how they shrink. Now think about the monomers. The beta tubulin here the monomer comes in the GTP bound way. This is the monomer. Okay? And there are always so many monomers around it, so the filament can find these monomers anytime. And they can attach here to the plus end, not to the minus end, because anyway the minus end is fixed on the MTOC. Okay, now we are talking about two speeds here. The first speed is the addition of monomers. And second, because these are GTPases, the second is the hydrolysis of GTP. Now, which one is going to be faster? In this case, the addition of the monomers, so the number 1 is happening faster. So before this GTP is hydrolyzed, new monomers are added, so it is getting faster. And this increase in size is very fast. This is why at the plus end, there is a structure called GTP cap. GTP cap is the piece of microtubule which contains the GTP, so they are not hydrolyzed yet. Of course, this is not a stationary structure. This is quite transient. As long as the microtubule is growing fast, you're going to see this GTP cap, and GTP cap has a very nice addition. It increases this monomer to monomer non-covalent interaction. So when it is bound to GTP here, the interactions between the monomers, they are stronger. So it is going to keep the keep the microtubule in a more stable way, so it will keep growing. So if the interaction between the neighboring monomers are not strong, what is going to happen is that it is going to peel off like banana. Okay? There are protofilaments here. And of course, these protofilaments are kept together by the non-covalent interactions. If the non-covalent interactions are weak, it is going to peel off this way, that we also call catastrophe. Okay? And then it will strongly depolymerize. So this is a growing microtubule. This is a polymerizing microtubule. And it has a GTP cap. If the hydrolysis of GTP is faster than the addition of the monomers, it's not going to have any GTP cap, and it is going to shrink, also called catastrophe. Okay? And it will simply depolymerize. So from the polymerized filament, you're going to have the monomers instead. Okay, here, because the GTP is hydrolyzed faster, there is no GTP cap anymore. So the interaction between the monomers are weaker. So they will start to detach into protofilament. And once these protofilaments are free, releasing of this GDP bound tubulin is quite easy. So next, they will be added GTP and they will be used as monomers in the cell again. And of course, when you check the cell, there is the microtubule organizing center. From this microtubule organizing center, the microtubules are growing. So there are some polymerized ones, plus there are lots of monomers. So the cell has to keep a balance of polymerized microtubule and microtubule monomers. If there is no microtubule monomers that is ready to be used, it cannot react fast to the coming signal. If there are too much microtubule monomers, and if the microtubule polymers, they all depolymerize, it would not be able to keep the shape stable, so it's again not good. So there is a balance of polymerized form and the monomer form, so it's coming and going back between them. And at the end, it is a continuously energy spending mechanism, but it has to be that way. I mean, we are kind of wasteful organisms. In order to keep alive, we have to keep dynamic instability. So what happens if you disturb the dynamic instability? Okay, whatever disturbs the dynamic instability is actually a poison. Okay, it's a toxin. But some of the toxins, we know that they are used as drugs. They are used for medical purposes. For example, Taxol here, it is used for breast cancer, against breast cancer. This is a chemotherapeutic agent. There's also the real name is paclitaxel, by the way. It stabilizes the microtubules. So the microtubules stay in this GTP cup version and they cannot depolymerize. Because they cannot depolymerize, okay, it sounds good. Maybe they're going to keep their shape forever. But remember, for cell division, all the polymerized microtubules, they have to depolymerize first and then they have to establish this mitotic spindle. Okay? If they cannot depolymerize, they cannot establish the mitotic spindle. And even if they can, in order to pull the chromosomes to opposite directions, they have to depolymerize. If they cannot, simply the chromosomes will not be pulled to the two sides. And just because the microtubule is disturbed, the cell division will not take place. So no microtubule dynamic instability, no cell division. No cell division means no tumor. Okay, it sounds perfect. You just block the cell division and everything is perfect, but unfortunately not, because it blocks any kind of cell division in our body. And we know that some of the cells, they have to divide frequently, like the hair follicles, like the fingernail roots, like the intestine stem cells, like the skin stem cells. Paclitaxel has also the side effect of stopping these cells from dividing. So it kind of makes people weaker. Okay, but as you remove the drug, the stem cells, they recover and they continue dividing. Or culture. Well, by the way, paclitaxel, Taxol is. Oh, something is happening. Okay, I need to stop it.